chick gastrulation. Okay, so this is occurring on top of the yolk. You're forming this thing called, sometimes it's called a blastodisc, sometimes it's called, the main part of it is called the epiblast, which is the part that the embryo will form from. So on top of the yolk, you have a bunch of cells that have divided, they form this blastodisc, then they pumped fluid that made a subgerminal space, and then into that subgerminal space, you formed another membrane called the hypoblast. So the epiblast delaminated cells, other cells came in from the hypoblast, came in from the posterior marginal zone, and you form the hypoblast, and in between the epiblast and the hypoblast is the blastocele cavity. So hopefully that part's easy to understand. Now we're talking about the next step, where cells are going to come down from the epiblast and form something called a primitive streak. Okay, this is not gastrulation starting yet because this isn't forming the axes, this is just getting things ready. Okay, so I was trying to explain to you, if you take this photo and kind of rotate it so posterior is in your way, and you're looking at just the epiblast, right, so that's what this sort of demonstration was. You have the posterior marginal zone here, you have the marginal zone all the way around, the epiblast is in the middle. If you take this embryo and you rotate it 90 degrees, so posterior is in your direction, and you're just looking at the epiblast, you're looking at just the cells that are going to form the embryo, these are the epiblast cells. Some of these cells are going to ingress, those cells have this marker called HNK1, that's not a functional thing, it's just a carbohydrate that they have on the cell surface. Those cells, those HNK1 cells, are going to ingress into the blastocele cavity, but they're going to still stay on the extracellular matrix, so they're not going through the extracellular matrix. Every blastocele cavity we've talked about has extracellular matrix, this is no different. So those cells are going to ingress, and then they're going to kind of form this lump of cells that's called the rudiment. Right? So those cells that were part of the epiblast cells are then going to converge towards the midline and extend from posterior to anterior. So when they converge and extend from posterior to anterior, they end up forming something called a streak. So when you look at the embryo, there's this streak that's formed going from posterior to anterior. Right? Once they form that streak, then those cells are going to degrade the extracellular matrix. Once you degrade the extracellular matrix, the cells are going to start moving into the blastocele cavity. When those cells start moving into the blastocele cavity, that is when gastrulation starts. When cells are moving into the blastocele cavity, the same as it is in sea urchin, gastrulation starts when cells are moving into the blastocele cavity, and basically the same as in xenophis, where you have the endoderm cells kind of moving around, and then the bottle cells come in, everything moving towards the blastocele cavity. Okay, so that's another overview. Which kind of takes us back to this diagram that the textbook has that has a lot of things going on. It's a little bit confusing. I hate it, but let's try to explain what the heck they're trying to talk to you about. So let's just start off on this side, say dorsal and ventral. So you're looking at the top, you're looking on top of the embryo with the dorsal view, and you're looking from the bottom with the ventral view. So let's start with the dorsal, which is hopefully the simplest. You're looking down on the embryo. So on the outside is this gray area, that's the marginal zone. In the middle is this light area, that's the epiblast, or the area pellucida. So you have the area opaca, the area pellucida. One side is thicker. Here they've drawn it red for some reason, so this is posterior. Okay, so this is just a normal embryo. You have an area opaca, which is the marginal zone. You have the area pellucida, which is the epiblast. And then you have a thickening on one side, which is the posterior marginal zone, so you know where posterior is. So when you're looking at the top of the embryo, on the dorsal side, eventually you see these cells that are going to be forming. These are those HNK1 cells that are going to be ingressing down into the blastocele cavity. Those cells will then converge and extend. So when you're looking at the top of the embryo, you'll see these cells come down from the epiblast, and then they will converge and extend, so they're moving from posterior to anterior. So that is the primitive streak. It's going to move from posterior to anterior. What else is this thing trying to show you here? This is trying to show you that if you're looking on the ventral side, so if you're looking from the bottom of the embryo, you'll see that eventually you're gonna form a hypoblast. So first you don't have a hypoblast, and then eventually you form a hypoblast. That hypoblast forms because cells are delaminating from the epiblast, and cells are migrating from this posterior marginal zone. So they don't really talk about how the hypoblast is forming. They mention